Hello, everyone. My name is Alessandro Busetti, and I am one of Metabolon's field application scientists. And as a scientist, I do consider myself very fortunate to have the opportunity to work on a plethora of different projects. In fact, the Metabolome represents the endpoint of the central dogma of biology. That is, all the chemical reactions that translate gene expression into protein expression and ultimately regulate our metabolism. In fact, metabolomics and the metabolon provide the most proximal output to a phenotype. Now, as many of you, I am currently working from home, and although metabolomics is a very powerful tool for different research applications that can be studies on aging, agriculture, the identification of biomarkers, bioprocessing, cancer and oncology, infectious disease, kidney, liver disease, studies on the microbiome, on neurodegeneration, and in general, pharma and R&D, I can't deny that my attention is currently focused on the concrete contribution that metabolomics can provide to research efforts during this global pandemic. So I decided to share a classic case studies on the rabies virus, and specifically on how metabolomics was able to identify key biomarkers for infection, key biomarkers of disease progression, evaluate the effects of vaccine treatment, and identify biomarkers of therapeutic response. I sincerely hope this very short presentation can provide some ideas and tracks to anyone who might be involved in virus-related research, but please do not hesitate to contact us if you require or want to discuss further anything related. So, as I was saying, the case study I was keen to present actually resulted in a publication that was published in the journal PLOS ONE in 2014 called Limited Brain Metabolism Changes Differentiate Between the Progression and Clearance of the Rabies Virus. And in this study, the metabolic profiles of the central nervous system were examined from rabies-infected mice that were either mock-treated or received post-exposure treatment with a single dose of the live recombinant rabies vaccine, Trigas. Central nervous tissue harvested from mock-treated mice at middle and late stages of infection revealed numerous changes in energy metabolites, neurotransmitters, and stress hormones that correlate with the replication of viral RNA. Although the large majority of these metabolic changes were completely absent in the brain of Trigas-treated mice, most likely due to the strong reduction in virus spread, Trigas treatment resulted in the upregulation of the expression of carnitine and several acylcarnitines, suggesting that these compounds are neuroprotective. The most striking change seen in mock-treated rabies-infected mice was a dramatic increase in the brain of serum corticosterone levels, with the latter becoming elevated before clinical signs or loss of body weight occurred. The researchers in this article speculated that the rise in corticosterone is part of a strategy that the rabies virus uses to block the induction of immune responses that would otherwise interfere with its spread. In support of this concept, we show that pharmacological intervention to inhibit corticosterone biosynthesis in the absence of vaccine treatment significantly reduces the pathogenicity of the rabies virus. Our results suggest that widespread metabolic changes, including hypothalamic pituitary adrenal axis activation, contribute to the pathogenesis of the rabies virus, and that preventing these alterations early in infections with PET or pharmacological blockade helps to protect brain homeostasis, thereby reducing the disease mortality. As I said, this brief presentation aimed to highlight the possibility to identify biomarkers of disease progression, such as the corticosterone, and these can be translated into druggable targets, as we see here with the use of metirapone, an inhibitor of corticosterone synthesis. So, as I said before, I hope this small, brief presentation can provide you with some insights and perhaps also quench a bit of your curiosity during this unfortunate pandemic we're all living. I do invite any of you who wants to know more to please contact Metabolon. Thank you all for your attention.